Hello, welcome, welcome. I'm not sure if anyone's here yet. Yesterday we did this. We had like loads of people in the chat already throwing topics at me for revision, but today we don't have that. So I'm not sure if anyone's here. Maybe everybody is busy revising. But yeah, let me know if you're here, if you've got any questions. The main purpose really was to show you guys, if you didn't get a chance to see it yesterday, was to show you the My Resources section on the My Edspace website, which is brand new and totally free. Hey Chinwendu, oh yes, hey Catherine. So I'm not sure whether you guys came yesterday. I don't remember seeing you. Um, but yeah, that was kind of the main purpose, was just to kind of hop on, have a little chat, how you're feeling about paper one, any questions and stuff. But it was really to show you all these free resources that you can be using for your revision over the next couple of weeks. Are statistical tests tested in paper one? Not as frequently, but they could be, Naira. Like you might have like some standard deviation on paper one. You might have to make a comment on like the significance on paper one. But like chi-squared, like specifically chi-squared often comes up with genetics, which is paper two. Just trying to think, when does Spearman's rank often come up? It might be to do with like ecology, which is more paper two. But it could come up on paper one because it's like a math skill, right? So it could come up anywhere. Can you explain the eyepiece graticule? Yeah, paper one is like year 12 content, but obviously like the math skills, practical skills, kind of there's like a theme throughout all three papers. But you're more likely to get specific statistical test questions on paper two or paper three as well. Do you need to learn about cardiovascular disease for the exam? Not specific examples, no, because that was actually taken off the specification. I think lung disease and cardiovascular disease were taken off the specification in 2016. So you don't have to know specific examples and like causes and symptoms. However, as we know, they could give you information and data, or, there's been an investigation or an experiment, and you might have to figure things out on cardiovascular disease, if that makes sense, but you don't have to like revise content for that. No. You've read the notes on Eyepiece graticule, but you're still struggling to understand it. Right, Chimwendu, one thing with Eyepiece, right, the Eyepiece graticule is the little ruler that goes in the eyepiece, but the divisions mean nothing. They don't have a set value because the value of those divisions will change depending on which magnification, which objective lens you're using. So you need to be able to say something like, using the stage micrometer, calibrate the eyepiece graticule. Because the stage micrometer is a little ruler that goes on the stage. It's on a slide and it goes on the stage, and that has divisions of known value. So you calibrate your eyepiece graticule using your stage micrometer, which all that means is you line them up. So you line it up so you can see your eyepiece graticule on your stage micrometer. And if your eyepiece graticule, I don't know, 10 divisions are equal to 1 micrometer on the stage micrometer, then you can figure out that one division on the eyepiece graticule is equal to 0.1 divisions on the stage micrometer. And there is an example on the study note, which I can show you, but I think you've already seen it. But yeah, the phrase is you use a stage micrometer to calibrate the eyepiece graticule because the stage micrometer, the divisions, so you can figure out how many eyepiece divisions fit into one of those divisions, and therefore how much an eyepiece graticule division is worth at that magnification. But you'd have to recalibrate it for each magnification that you use on the microscope, each different objective lens. I don't know if that's helped you. What would you say to prioritize today? Oh, should we memorize all the past paper? I wouldn't bother memorizing. I think you should prioritize topics that you find the hardest. And I would prioritize things that are hard in general, like prioritize meiosis, prioritize the immune response, prioritize mass transport in plants and animals. I mean, I'm listing like a whole bunch of topics now, right? Like, you can use the study notes on my Headspace, which I will show you in a sec. You can use those, like go straight to the topics that you find the hardest. Like, if there's a topic that you think I would hate a question on that, go to that study note, read it, and try the TikTok question and the exam question at the end. And then you can always access more questions on that topic online, right? Can you explain tissue fluid briefly? Catherine, have you seen, can I just show you this first? Rather than me trying to kind of teach a big, difficult topic like that in a short time, I'll just go to the study note and see, you can let me know whether you've already seen it. And you can let me know what you don't understand specifically, maybe. Can we go through a cardiac cycle question that uses a graph? Have you seen, were you here yesterday, Naira? Because yesterday on YouTube, I posted a video on the cardiac cycle with a graph. So if you've not seen that video, it's the graph where there's like pressure changes on the y-axis, and it shows you the pressure in like the atrium, the ventricle, and an artery. And that was posted yesterday on my YouTube channel. And today, actually, I've just posted a meiosis video, like quite a long, like 20-minute meiosis video. So if that is one of those topics that you would hate a question on and is quite likely to come up, then you can watch that video as well. Tissue fluid was what we were saying, wasn't it? Let's find it on here, just so you can see what the study notes kind of look like and whether you think they will be useful. Why can't I remember where it is? Here we go. Mass transport in animals, tissue fluid. Yeah, so the good things about the study notes is however you learn, like there's something for you. So you've got recall questions at the top just to check your prior knowledge that you might need to access the note. Then you have got a video explainer, which is like a little mini lesson. This one is from Joe. Some of them are me, some of them are Joe. Then you've got a full set of notes. And what's good at this point, I suppose, because we're so close to the exam, is you can go to like describe how tissue fluid is formed and how it is returned. And then we've given you like a perfect model answer that we've written based on past paper analysis and looking at mark schemes. So I can't remember who it was now that said tissue fluid, but I mean, you can quickly watch the explainer video. You can put it on double speed. You can read the notes or skip straight to the question and like learn the model answer. Because at this point, I understand we are running out of time. But that, and then there's another question. So you could have a go at this question, look at the model answer, and then play this question, pause it, have a go at answering it, and then watch my solution. In fact, there's two practice questions there for tissue fluid. Hi, have you covered evidence for mass transport? On here, yes, we have. So there's absolutely everything on here. So we're currently in the mass transport in animals section. But if you go to the mass transport in plants, experimental evidence, including tracer and ringing experiments is there. And it's just loading now. And everyone is kind of the same. So there's always recall questions, which kind of about like the prior topic or maybe GCSE knowledge that you need. There's always an explainer video. This one, you've got evidence for cohesion tension theory, evidence for mass flow hypothesis, translocation. So you've got like radioactive tracers, ringing experiments, aphid style experiments. There's always key terms, exam tips, and then you've got another practice question there at the end. So yeah, there's everything on here. So the reason I'm kind of showing you this, I showed everyone last night and I'm showing you again today. Obviously, we're really short of time now for paper 1. You've got a little bit more time then for 2 and 3. But on the My Headspace website, these are all free. If I haven't said it, they're all free. It's the whole specification for AQA, but many of them are going to be useful for OCR, right? Because lots of the content is the same. So at this point, you can just jump in, go straight to the study note that you need extra help with, watch the videos, read the notes, and try the questions. Okay, there's nothing here that's paid for, by the way. It's all free. But yeah, also check out my YouTube, because cardiac cycle, a lot of students last night were asking me about that, and I'd literally just posted a video on YouTube. And meiosis, a lot of questions were asking about, and today we've posted that video. Evaluation and application questions. You keep saying things that are not worded like they are on the mark scheme. I mean, the mark schemes are quite strict. With application questions and evaluate questions are different, right? Application basically means you are taking your knowledge of a topic and applying it in a new context. So the same rules apply as with any straight AO1 questions. You still have to use your knowledge, you just have to apply it to a new situation. So your knowledge has to be sound, you have to use correct scientific terminology, you have to have the same number of points as the number of marks. Make sure, like I always kind of say, each point you make, you should have some key scientific vocab, like in each bullet point. Like, I don't know, osmosis, water potential gradient, you know, that kind of thing. Evaluate is different, and evaluate, I mean, I find them easier than application, because usually with evaluate, you kind of know if it's this many marks, I need to split it and do, you know, if it's 6 marks, 3 pros, 3 cons. If I can't do that, I'll also get away with maybe 4 pros and 2 cons, or 2 pros and 4 cons. And what you're evaluating will differ depending on the question. So are you evaluating an experiment, a method? Are you looking for good things about the method? Like, yes, they had a control group, and yes, it had a large sample size. And then are you looking for bad things about the method? Like, I don't know, the sample size was too small. They did this on rats, not humans. They only studied the effects for 8 months, so we don't know what the long-term effects are going to be. Like, it might be that kind of evaluate. So look at the method and pick out the good things and the bad things. It might be evaluate the data, which is different again, isn't it? It might be that you've got a graph or a table and you're saying, yes, there seems to be a positive correlation, but it's a weak positive correlation, and there are many outliers, and correlation doesn't prove causation. There's other factors involved. Some of the data overlaps with other data. Like, they're very different, but it's always like for and against, and you have to use the information, use the method, use the graph, use the table. I hope that's helped. Don't be scared, like don't lack confidence, because if you go in thinking I can't do those questions, you're just going to panic when you see it. Have a go. Even if you can't get all the marks, you might pick up 1 or 2, and those 1 or 2 marks might be the difference between a B and an A. How different A levels are to GCSEs. I think biology is quite a challenging A level. Not going to lie, not going to pretend, but I think all sciences, particularly science, maths, very challenging A levels. I think what's nice about biology A level is it does build on your GCSE knowledge. So there's always something you already know from GCSE, and we can then gradually build it up. It gets progressively harder. So it's not like you're thrown in at the deep end immediately. Like the start of year 12 is okay. Year 12 gets harder, then year 13 gets a little bit more hard. So it's gradual. But if you enjoy it for GCSE, you'll probably enjoy it for A level. That'd be my advice. If you enjoy it, then take it. If you get a 6 or above in your GCSE, you should be fine. When is the cram session? Tomorrow, Anaya. So cram is tomorrow 4 till 9. If you're AQA, you'll be with me. If you're OCR, you'll be with Joe, just to make sure you have the right teacher. So OCR is with Joe, because obviously there are differences. So we're running 2 separate ones. I've done it a little bit differently this year. Like I've planned, well, I always go over. I do always go over time. But I've tried to limit my teaching to like 4.5 hours. So we can spend half an hour at the end just doing like whatever you guys want me to do. Basically, I'll have some blank slides and I'll be like, right, what else do you want me to go over? And I'm hopefully going to have some time to do that. Oh, manage the time during the biology exam. Catherine, on paper 1, I would do the last 3 questions first, because they're going to be the easiest, most guaranteed marks on paper 1, because they're the long answer questions. Do those first, then go back. Just keep checking the time. If there's a question that you know, right, it's only 2 marks and I've already spent like 8 minutes on it, leave it. Don't miss out other marks that might be easier because you're spending too long on a really hard question. If you've got time at the end, obviously go back to it, but just, yeah, try and be a little bit thoughtful about that. Any other questions? How do you, let me show you. Right, at the end of this, there is it in the exam skills section? I'm just trying to think where it is. So there's an exam skills section here as well, which we've split into a few different parts. This will probably get added to over the years. It'll be here for a long time, but we'll probably add to it. But yeah, it's already got quite a lot in here. So you've got choosing a statistical test. I'm just checking because I'm sure I put in a table. Not that one. That's about choosing the right statistical test. So it wasn't that one. Interpreting results from, so we've done a separate one on interpreting results from a statistical test. So it's probably on that one. And yeah, we've covered everything. So AQA often give you a p-value. So we've talked about how to interpret p-values, but they might give you a table with critical values in. So we've can't speak, also done how to compare the test statistic to a critical value. And yeah, here you go. So here, for example, you've got degrees of freedom for the chi-squared, the student t-test, and the Spearman's rank. So you've got, I mean, you don't have to learn the equations, right? You're not going to be calculating them, but you should know when to choose each test. You should know how to write a null hypothesis. And degrees of freedom usually comes up for chi-squared, by the way, because chi-squared is the one that links to genetics. So for example, if you've got 4 different categories, like 4 different phenotypes, then degrees of freedom would be 3, because it's the number of categories minus 1. I don't actually think I've seen them ever ask for any of the others, but you have got them there. You've done all the A-level papers. Would it be good to do S papers too? Yeah, Tia, for sure it would. Yeah. AS papers as preparation for paper 1, I think brilliant. If you've done all the A-level papers, then yeah, definitely. Do all the practice you can get, says Anaya. Yeah, for sure. The more, the better. And a lot of the AS questions, you might find them slightly easier. Yes, because if you're doing AS, obviously you don't have any knowledge of year 13 yet. So they won't be as challenging, but a lot of the questions are very similar in terms of difficulty. So definitely. Okay. Any other questions? Is there any other study note? I just want to show these study notes. Like, I'm so proud that we've finally got these published. I'm like wanting to show them off, I suppose. They are hopefully going to be really useful. I know you guys haven't got much time to use them now, but don't forget, after paper 1, go back to them. You've got a little bit more time then for paper 2, and you can go straight to the topics that you really struggle with. Yeah, so they're all free. I'm just going to show you, in case you weren't here at the start. This, by the way, is the My Headspace website. So you just go to My Headspace on Google, and the way to access them is just click on My Resources, A-level biology study notes. And then you can go to, it's following the AQA specification, but as I said, a lot of the topics are common. So if you're OCR, AQA, like everyone has to do DNA replication, right? Or transcription and translation. So you just go to whatever you want and open it up. So scared for paper 2, or yeah, these resources will be available next year. Yes, they'll be there. They're just on the internet. So they'll be there for a long time, hopefully. If anything, they'll just get better and better. Are they, Aaliyah? Oh, it's Alien. Yeah, probably. I didn't know that she was live as well. But yes, Make sure you use them. Let me know. You can always message me on social media. Let me know how you find them. You can even suggest improvements. That would be amazing. But yeah, they will be there for, if you're in year 11 now, they will be there for you if you're going to do A-level biology. When we get the critical value, we just ignore it and only consider the given p-value. What do you mean, Chinwendu? AQA do, like, sometimes they just tell you the probability is less than 0.05. That is them telling you that the probability of the difference occurring by chance is less than 5%. But sometimes they don't tell you that, Chinwendu. You have to compare the calculated test statistic to a critical value. But when you look up your critical value, you have to look at the 5% probability column, and you have to look across at the correct row for your number of degrees of freedom, and you compare the result of your chi-squared to the critical value. And if that's what you're doing, you want the result to exceed the critical value in order to be significant, a significant difference. But use the study note will explain that better, right? Better than I can in a short time. So go to interpreting the statistical test result and look at the critical value section. Okay. Inaya, that you can kind of ignore that. That just means that like 95% of the data falls within plus or minus 2 standard deviations. But kind of just, they just say that. You don't really have to reference that. You just talk about like standard deviation. Obviously, it shows the spread of data around the mean. You should know that. You should know that if there's a big standard deviation, it means there's a lot of variation in the results. If there's a small standard deviation, the results are more similar, so they're more repeatable. And then often with biology, it's about the overlap. Do the standard deviations overlap? If the standard deviations overlap, the difference is likely not significant. If they don't overlap, then it's probably a significant difference. So you just kind of look, they'll just give you the standard deviations like in a table or on a graph as error bars. And then if they do that, they want you to use them, right? So they want you to look for overlap or no overlap. PSY. Yeah, we've just talked quite a lot about that. But PSY, all I'm going to say is go to... It's not on there now, is it? Where's the... Let me just click into one. There's an extra... Oh, here, exam skills. Can you see here? We've just talked about it, so I'm not going to talk about it again, but there's choosing a statistical test, and this is the one that you want, PSY. You want interpreting the results from a statistical test. It goes through everything. I have written it. It's in the way I would tell it you right now anyway. So yeah, you can have a look at that and go through what the p-values mean. I also think I've literally just posted a video on my TikTok on p-values, like a really basic breakdown of exactly what it means, like really simplified. What do the p-values mean? So maybe check that out as well. In fact, it's this one. I've put it into the study note. It's this one here, this video that you want to watch. Yeah. You could be asked to draw a scientific diagram. Yeah, biological drawing, you mean? Yeah, you could. It's come up a few times. You've had to do one of a mitochondrion before. You've had to do a tissue map of the tissues in a leaf before. And remember, if it's a tissue map, they don't want you to draw individual cells. They just want like the outline of each tissue. So on the leaf one, they wanted like a band for the upper epidermis, a thicker band for the palisade mesophyll, another thick band for the spongy mesophyll, and then a band for the lower epidermis. You don't draw every cell, you just draw the outlines of the tissues if it's a tissue map. Okay. Do you have a service where you make essays? No. But there are some on my YouTube channel, like example paragraphs, essay tips, videos on stuff like that. Chinwendu, I don't understand what you're saying there. If the probability, I don't know what you mean by value. If the probability, if you're told the probability is less than 0.05, that means the probability of the difference occurring by chance is less than 0.05, which means it is significant. But I don't know what you mean by the value. Chinwendu, and you're mixing up your words a bit there, like reject probability. I'm not sure what that means. You need to look at this study note. You need to look at this, Chinwendu, please. Watch this first video and then go through all these notes. Yeah, make sure you've got it clear because you're starting to mix things up. And I know that you did used to have quite a good handle on this. OCR. Yeah, OCR biology. You can join the cram course with Joe tomorrow. Mahi, is it? The cram course. Joe's doing a cram course 4 till 9 tomorrow. There's still time to sign up if you haven't signed up yet. Okay. How do you find these notes? Yeah, this is just the My Headspace website, Chloe. Can I go over topic 2 cells? I can't now. I mean, that's a whole module. That's like 8 weeks of work. But for sure, here you can, because you just go on to cells. We've split it into cell structure. Like, look at all that. That's all cell structure and studying cells. Then you've got all your cell division. Then you've got all your transport across cell membranes. Then you've got all your immune response. So you can go to whatever you want to learn about on module 2 cells. You'll notice that we've also got all the required practicals like root tip squash. We've got, what other practicals have you got? The heart dissection, required practical. Like they're all, every practical is on there. Even practicals that aren't required practicals like the potometer and how to use a potometer is on there. And it's all in order, so it should be easy to find. Yeah, I'm doing GCSE cram for bio on Sunday. I think that's Sunday 4 till 7. Can we still do your cram course? Yeah, AQA. I'm doing the AQA cram course tomorrow and Joe is doing OCR. And if you do sign up, by the way, you do get all 3. So it's only £20 and you get the cram course tomorrow and the one before paper 2 and the one before paper 3. It's not an extra payment or anything. You get like the full 15 hours for just £20. So it is quite a bargain. Yeah, Anaya, we tried to write them without any waffle. Like we tried to keep them like, this is what you have to know. And we did use our experience as examiners to kind of write them in like mark scheme style speak. So they're not really long and waffly. It's very to the point, it's very concise, and it's got all the correct terminology that you need. When we write about humoral immunity, do we need to talk about how they become APCs? No, I don't think you do. You just talk about, obviously you need to say that the B lymphocytes are activated by the T helper cells. That's important. So you have to mention the T cells. B lymphocytes are activated by T helper cells. Then they divide by mitosis. Then they differentiate into plasma cells and memory cells. And the plasma cells are important because they're the ones that produce the specific antibodies. Triple bio cram course. We talk in GCSE, Daniel. You can stay for 5 hours if you want, Daniel, for paper 2, because you're going to have 3 hours with me for combined and triple, and then triple are going to get an extra 2 hours if they want to stay for an extra 2 hours, because there's a lot of extra triple content on paper 2 GCSE. So if you want to be with us from 4 till 9, you can be. How are they activated? I mean, for AQA, you don't really explain it fully ever, but what happens is the B lymphocyte has receptors on its surface, yeah, like antibodies acting as receptors. They bind to a foreign antigen, they present the foreign antigen, And they are also activated by T helper cells that release chemicals called cytokines. But you can just get away with saying that they are activated by T helper cells. The correct B lymphocyte is activated by a T helper cell. Meiosis Chloe, literally just posted on YouTube. Get on it. Big video on meiosis, including crossing over and independent segregation, just was posted today. Nice. Right, guys, I'm going to love you and leave you. I hope you are all going to use these notes. I do recommend them. Obviously, myself and Joe have written them. We do have many years of teaching experience and experience as examiners, so hopefully you're going to find them really great. GCSE ones coming soon. Not available yet, unfortunately, Daniel. But they will be there at some point next year. Would you, Anaya? Yeah, that'd be good, wouldn't it? But you have to be careful not to just give GCSE answers for that. You'd have to try and work in some extra A-level stuff for that one. Thanks, Chinwendu. Yeah, thank you for joining me. Use the notes. You can always let me know how you find them. And if you've got any advice for how to improve them, that would be useful as well. And if I don't see you in the cram course tomorrow, then good luck for Thursday. But hopefully I'm going to see a lot of you there. 4 p.m. It's on YouTube. The link is in the description of my YouTube channel, I think. So you can sign up still if you haven't already done so. The link's also on my TikTok. Yeah, I think so, Catherine. Bullet points. As a marker, I prefer it when they write in bullet points. As long as the bullet points aren't so brief that they don't get the marks. Bullet points help you to structure your answer. They help you to hit the number of marks that the question is worth. So I would use bullet points. Yeah, we're doing loads of questions, Chloe. Questions from lots of different parts of the spec, lots of different styles, but also like focusing more on questions that I think are more likely to come up. Not really predictions, but just like based on past paper analysis, what's more likely to come up. We're going to do a few more questions on those topics. See you Sunday, Daniel, and I'll see you tomorrow, Chinwendu. Thank you, everybody. Have a lovely evening. Thanks for joining me. I will let you go, and I will see you tomorrow, hopefully. If not, good luck for Thursday. Bye.